and welcome to this lecture on production facilities for AAV, adeno-associated viral vectors used for gene therapy. This lecture is part of a lecture series at the University of Aachen in Germany for cell and gene therapy. And my name is Jørgen Magnus, and I'm head of the biochemical engineering in Aachen. So we're going to look at the use case here. We're going to look at the specific process that is going to be hemophilia A. And we will see how we can design a facility to produce material for 4,000 patients per year. So that is the capacity. So imagine if you're a process engineer and you get a message from your commercialization department that you need to provide material for 4,000 patients per year, and then you get the task of designing the production facility for that. So that's the exercise that we're going through today. One question that you have to ask yourself is, what is the scope of this facility? And here we will assume that this facility produces both the drug substance as well as the drug product. The difference is that drug substance is really just active pharmaceutical ingredient purified and in a buffer, in a defined buffer. But then you also need to fill that in vials and package it and send it to the hospitals, and that's your drug product then. So the finished vials, that's the drug product. Often drug substance and drug products is done in different facilities. In that case, you would have a separate facilities where the filling machine is located. But in this case, it makes sense to have both those functions in one facility, where the filling machine can be also rather small, because we're not filling that many vials per year. And so it's okay to fit it into the same facility as the drug substance facility. So the first thing that you have to think about when you decide the facility is how big are your bioreactors going to be, and how many bioreactors are you going to have in the facility. Because once you know that, you can calculate everything else based on the size of your bioreactors. So the whole equipment for downstream processing depends on the size of the bioreactors, and then the buffer preparation and the media preparation, also the utilities, all of that is then related to the size of the core process. And around that, again, you would build the facility, the building that fits that can house the production process. So the first step is to look at the bioreactors, and here we will show how you can get from the assumption 4,000 patients to a size of a bioreactor. So looking at these calculations, we have some assumptions that we need to make, or some data that is the basis for our calculations. So we have the 4,000 patients per year. Then we have the doses per patients. This is given in virus genomes per kilogram of patients, because we're talking here about the systemic injections. Hemophilia A is then a systemic injection. So you would adjust the dosage to the weight of the patient. And we assume in this study that the dosage is 10 to the power of 13 virus genomes per kilograms. But then you also need to make an assumption about the average weight of the patient, which in this case is 60 kilograms. We have assumed that we will treat mostly adult people, but also some children, and then the average weight of the patient could be something like 60 kilos. That gives you the dosage per patient. And then you have to consider losses in the processing. So if you're calculating yourself backwards from the amount of final product towards the bioreactors, then you will have to consider the loss in the filling operation and the QC sampling. And this can be as high as 40%. So in the filling machine, you will have sterile filters that you cannot flush, because then you would dilute your product. And so you have significant losses in your filters, also in your tubing and your bags that you're using in the filling machine. QC sampling can be very significant because you will have to take a significant amount of samples to check all the quality parameters that you need to check to be able to release the product. And even the fact that you need to fill a little bit more liquid in the vials than is actually needed, just to be able, for the doctors to be able to aspirate the 2 milliliters, for instance, you would have to fill maybe 2.2 milliliters in the vial just to be able to avoid air in the syringe. So that gives you a 10% loss. And in total, we assume here 40% loss in that operation. Then in the downstream processing or the downstream purification, you can have as much as 70% product loss. This is the loss in all the chromatography steps. It's in the filtration steps. The depth filtration will have significant loss. Also the ultracentrifugation, if you're using that to separate empty and full capsid, will also have significant loss, and in total it can be as high as 70%. Bioreactor runs aren't always successful, and there can be technical issues, or there could be, in the worst case, a contamination to your bioreactor, in which case you will have to throw away all the contents of the bioreactor. So assuming a 90% success rate or 10% unsuccessful runs is actually quite a good performance in a facility. The titer in the bioreactor for the most common serotypes could be as high as 10 to the power of 14. So if you're producing maybe AAV8 or AAV9, that is a reasonable titer with a good production cell line. 20 bioreactor runs per year. Remember that we said that one bioreactor runs for 7 or 8 days, so you will have 4 days of growth phase and then 3 days, for instance, of production phase. But then to those 7 days, you need to add at least 1 day before for preparation and 1 day after for harvest, where you're also occupying your bioreactor. So let's assume 10 days per run. And then if you run 20 runs per year, you will have 200 days utilization of a bioreactor per year, which is quite a good number. There will always be some downtime between the runs. You might also have to shut your facility for a couple of weeks to do maintenance. So 200 days of operation of a bioreactor is very good, which will give you then the 20 runs per year. We are assuming in this study that we're using single-use bioreactors, and the standard size for single-use bioreactors in cell culture in large-scale manufacturing is 2,000 liters. So this has been a very established size, and this is also the assumption for this study. So now we can start calculating how much product we have, and this is just simply the number of patients times the dosage per patient. So that will give you the virus genomes that you need to produce per year. And then we can start calculating the losses. So just considering the loss in the filling, you would just divide by 0.6 then, because you have 40% loss. So that would be, if we keep the Excel sheet alive, we can have we can calculate the product that we will have to have before filling, considering the loss in the filling. And then the product that we need to have before the purification and before the loss in the purification is just this number divided then by 0.3 to count for the 70% loss. So that's just this number here. And then considering the 10% unsuccessful runs, again, we divide by 0.9. So subtracting here the 10%. And then we can calculate the total volume on bioreactors, which is essentially the product that we need to have in the bioreactors divided by the titer. So the virus genomes per liter will give us the number of liters that we need. So that is this number here. 148,000 liters per year, that's the processing volume that we need, and then per batch, dividing by the 20 batches per year, will give us the amount per batch. And then we said one bioreactor is 2,000 liters. So dividing by 2,000 liters will give us then calculate the value of 3.7 bioreactors, which means that it would make sense to design the facility with 4 bioreactors. So that's the result here. So 4 bioreactors. is the conclusion after these calculations. So if that was a bit quick, then it doesn't matter, because the point was just to show the assumptions that is behind these calculations, and they aren't very complicated. There's also different ways of doing it, of course. But we should know that these assumptions aren't very accurate, and this is often underestimated by people who are planning facilities, because imagine if you don't get your market share of 4,000 patients per year, but maybe it's only 2,000, or maybe it's also 8,000. In that case, you need half the size of your facility or twice the size of your facility, just because the market projection changes. So and the same goes for all the other parameters. Dosage, of course, very important. This can vary a lot. If you're treating, if you have a different type of treatment where patients weigh a lot less, for instance, imagine if you're treating newborn children and they weigh maybe a tenth of what you have assumed here. It means you're off by a factor 10 in your calculations. So this fact has to be appreciated, I think, and it makes sense to calculate different scenarios. It makes sense to consider different therapies that can be produced in this facility, and then you find a compromise of a sensible size of facility that most likely will give you the amount of product that you most likely will need. But it's impossible, of course, to make a perfect calculation here, given the uncertainties in the basic assumptions. So now we know the size of the bioreactors, and we have concluded with 4 bioreactors to be able to treat 4,000 patients. We mentioned that we're using 2,000 liter scale and single-use technology. Suspension culture is the main operation mode, but we also want to have the flexibility to also use adherent cell culture. That will be a very different type of production, but also at least designing the facility so that is possible makes sense to have this flexibility. Then the multi-product capability in a campaign-wise means that you can run one campaign of hemophilia A, and then you change to a different product in the next campaign. And this is possible because we're using single-use technology, and we want to use single-use technology for all the processing steps. So all the steps that has product is made in single-use technology, which means that the product only touches single-use equipment. And this is changed, of course, for every batch, which means that it's not that difficult to change from one product to another. You at least will avoid the risk of cross-contamination due to insufficient cleaning of your equipment, because you have actually changed, since in single-use technology, you have changed everything. All the surfaces that touches the product has been changed. And also you take certain other measures. For instance, you do fumigation of your production rooms, which means that you fumigate them with vaporized hydrogen peroxide, which disinfects all the surfaces, the walls and the ceiling and the floor, and the outside of the equipment is then disinfected by hydrogen peroxide, which also reduces the risk of a batch-to-batch contamination. What comes out of this is a rather modest size of a building, 1,900 square meter in footprint, 17.5 meters tall, and it has been condensed with one production level and one further level for storage and gowning, etc. Plus the HVAC level, of course, and also walk-on ceiling to be able to do maintenance and to organize the piping and the HVAC ducts. So maybe a note on cleanroom classification here. We have assumed that we can use grade D in the production level. So this way of classifying clean rooms, the A, B, C, and D, this is related to the EU GMP guide that uses these categories. There's also different ways of doing this. There's an ISO standard for clean rooms as well. And there used to be a US federal standard for clean rooms that is not in use anymore. and I think most people use the ABCD system. Essentially, they describe how many particles you will have in the air of a certain size. So as example, grade C, you can have maximum of 352,000 particles larger than 0.5 micrometers per square meter air. So it just specified how much particles in the air you're allowed to have. But it also distinguishes between operation and at rest. So the number I said relates to facility at rest, which is the state when you're not running the facility, but the air handling units and the HVAC is still running. But then in operation, there's a higher limit, typically a factor of 10 higher of the maximum limit of particles in the air. So that's what these grades are saying. They also, of course, give recommendations on colony forming units and the maximum colony forming units in the air, but this is recommendation and not strict requirements. Looking at the manufacturing block, we have the cell culture and the harvest, of course. We have the purification rooms. We have the drug product filling rooms. We have the room for inspection, labeling, and packaging. And after that, the product can leave the facility. And then we also included a room for plasmid production in the case that you're using a triple transfection process and you can produce your plasmid in that building. Which means that you don't have to buy them from an external company or produce them yourself in a different building. So that makes it a little bit easier. And then another room for cell and virus banking, because you might need, if you're using the HeLa system, for instance, in a production cell line, you have to infect it with the adenovirus, and therefore you need also virus bank. Utilities is reduced to a minimum. There's a WFI generation unit, so water for injection. But it has been assumed that we don't need clean steam generation here, or if you need small amounts of it, you might be able to take it off your WFI generation unit. It's assumed, by the way, that this facility is on established production site, which means that you have steam and water and electricity and other utilities available on the site, and also the services like wastewater treatment and central storage of consumables should also be available on this site. And we're also not considering the analytics laboratories, so they are also assumed to be in a different building than this. So this is just a production, essentially. And finally, it also has the liquid and solid waste inactivation. So what comes out of all this? It's a very modest investment sum of $102 million US, where the operational expenditures will be $28,000 US per patient, and 52 full-time employees working in the facility, not at the same time, but in shifts then. And the timeline, which we will look at the very last slide in this lecture, can be 30 months, including the basic design, and then up until the end of the operational qualification. So we will look at that a little bit later. First, we repeat this slide from the last lecture. This has been simplified a little bit, but it's just to show the different manufacturing steps that you have to consider in the facility. So we have the manufacture of infection and transfection solutions. We have the cell expansion in the seed lab, the fermentation where the main fermenters are located, also the pre-cultures, and then the purification steps, and in the end the filling machine. So these are the main steps to be considered in the process. And then we have the expression systems that we mentioned. We want to design the facility to be able to process all these different expression systems. The HeLa cells, which is a production cell line that needs infection with adenovirus. The insect cell line SF9 that is co-infected with 2 different types of baculovirus. And then we have the triple transfection, the most common expression system in HEC293 cells, where you use plasmids. At the end of it, we also included and tried to consider here a production process for lentivirus, just to have that as well. And lentivirus can be produced in suspension culture too, but it has been assumed here that we have adherent cell culture, just to be able to look at that production scenario too, which will be very different. I think the first three ones here can produce in very similar equipment. You have to, in the first two cases, you have to consider virus inactivation a little bit more than in the last expression, because in the last expression system, you're using plasmids rather than virus in the production. But essentially, it is very much the same equipment that can be used here. Looking at this equipment that has been planned in, we have the cell culture area with the biowave and the smaller bioreactors, the larger bioreactors that you see here, the 2,000 liter bioreactors, the depth filtration step. If we're running a HeLa production cell line, we also need to add the adenovirus, which we will do to the main culture here. And then we also consider the benzonase step, which is to remove host cell DNA, so the DNA that results from the cells, the production cells. And the benzonase is added to the bioreactor after the production phase, so that you can dissolve the DNA directly, and then it should be a lot easier to do the clarification steps, and your depth filters won't block so easily once you have done the benzonase treatment. Then we have the purification area with all the chromatography units and the filtration units. We have the filling room with the filling machine, also the final formulation, another ultra and dye filtration step. And then finally the inspection, labeling and packaging room. And here it's already mentioned biosafety level 2, because these areas with the frames where you have processing of virus needs to be designed according to the biosafety level 2 guidelines, which means you need to take some measures to protect the operators from a biohazard. And we will see later what that means in practice. But in principle, the most important assumptions behind the design of this process is the 4 bioreactors, 2,000 liters that we calculated. We can start by installing just one of them. For instance, if you start by providing material for clinical studies and you later want to go to commercial production, you can install the other 3 bioreactors one by one later, and then also upgrade your facility to a commercial facility. As we mentioned, the filling isolator is part of the final purification. It's an isolator in a class C room. And therefore it has to be an isolator, because an open filling machine would require a class B background, but with an isolator it's okay to have a class C. As mentioned, 10 days per bioreactor run, 20 batches per year, gives you about 50 patient treatments per bioreactor run, and the size of the filling machine is rather modest, with 50 to 100 vials filled in 2.5 days. So that is the basic assumptions of the process. Looking at the building, we put everything related to production on one level, and that's level 1. And in level 2, that's where people are entering. So people would enter from one side of the building, and then the materials could enter from the back side of the building. But essentially you can store materials down here. You can also have gowning areas or places where people that enter the building can change from their street clothes. The kill tanks that inactivates the liquids that are contaminated with virus is also located down here. Other types of utilities, WFI generator, all of that will be on level 0. And then you have the production level, and then you have this walk-on ceiling where you see the HVAC ducts, and there should also be piping there. And at the top floor, it's the air handling units that provides the HVAC for the production rooms. So that's what gives us the the design of the facility, and as mentioned, it's most of it in grade D here, because we went for a grade D design, which is maybe not common in these facilities. They're often designed with the classroom C. But in our study, and because we are using the principle of close processing, it's okay to design it as grade D, and the product remains protected because of the close processing. And also the single use contributes to that as well. So that's why it's possible to downgrade from class C to class D. Now, maybe coming to the most interesting part of the production facility, because imagine that people have entered on the ground level, and they will move then up to the production level on level 1. And there will be 2 different teams. So there will be a team that is involved with production, and this will be the clean team. So they have to have a different level of cleanliness. And then there will be a team that takes care of waste, and this will be the dirty team in a way, and they are completely separated in that building, so they're never in the same room together, and there's no contact between the 2 teams. So the clean team would come up through the stairs up here, and then they could go into an airlock and gown, and so one for men and one for women. And after that, they can move into this clean corridor here, where they can move all around the facility and have access to all the production rooms here. So that corridor is the clean corridor, or the supply corridor that goes around the whole facility and gives you access to the production rooms. So that's where the clean team would be moving in. And then, for instance, they can move into this area here, which is the buffer and media preparation area, where you have tanks that prepare all the buffers and the media for the process. And then you have storage bins here, located next to the walls, and they're serving the bioreactors that you see here. Here are the 4 bioreactors, and also the different chromatography columns and the filtration units that also need buffer in the purification room. So that's the buffer and media preparation room, and also the holding bags for that. And then we have the cell culture room. Here is the main bioreactors, and also the harvest area. We have the initial purification, which is the downstream processing, and then the product moves into the filling room, where there's a small filling machine here, and also the final formulation steps that takes place in this room. And this is then the classroom C, while the other rooms are classroom D. So there's the purification is D, and the bioreactor is D. This is also a D room with the buffer preparation. And then finally you have the inspection and labeling and packaging room, where you really check that the vials are intact, and you can label them and package them, and then you can move them out of the facility again through the corridor and back downstairs to leave the facility to go to the hospitals. And the process starts actually here in this room. This is the seed lab. So the vials are stored in liquid nitrogen in this small room here, and then brought into the seed lab, where you have the first shake flasks cultivation and the first passaging of the cells. And then when you have produced, for instance, 2 liters, you can move into the production room where the biowave is. And the seed lab is also a clean room class C. So the two C rooms here is the seed lab and the filling room, and everything else is then class D. There is in the cell culture area where the bioreactors are a separate small room for harvesting. And this is because it's difficult in the process to avoid spilling liquid on the floor when you dismount the depth filters that you're using for harvest. And so to contain those spills a little bit, it's done in a separate room, which is then also located right next to the airlock here that leads into into the waste corridor. So this central corridor here is a waste corridor. So all the used equipment from the cell processing, but also from the downstream processing, can then move into the waste corridor, and there it can be collected by the waste team. And the waste team enters the facility or the production level from the stairs here, and they don't need to go on because they're only dealing with the waste, so they can use the same. It's still going to be not their street clothes, but it's going to be the production clothes that they have changed into on the ground floor. And they will only move in this waste corridor here and collect the waste and bring it into this area here where you have a decontamination autoclave. So this is the return corridor, and then we have the autoclave for decontamination here. And after that, the decontaminated single-use equipment is then returned downstairs to the ground floor. Plasmid production and production of infectious solutions could take place in these two rooms here, so one room each for those functions. And that's essentially a very compact design of a production level that will give you all the functions that you need to produce the product. Looking at GMP requirements and biosafety requirements, these are often considered together. So close processing, we mentioned, is a major is a very important measure to take, first of all, to protect the product. So that would be the GMP aspect of it, that you protect product from, for instance, particles in the air, or maybe microorganisms that in the air, and when you keep the processing closed, you can very greatly reduce the risk of contaminations from the surrounding air, from the environment. And we won't go into all the details on how to do close processing because that is a very long and separate topic, but you will essentially use bags for almost everything, for bioreactors, for holding liquids, and connect those bags with tube welding or aseptic connectors. This is also important for the biosafety aspects because you're also protecting the operators of this facility from the virus in your product, and therefore it has both those aspects. It serves both the GMP aspect as well as the biosafety aspect. The unidirectional flow in the processing is also good in the sense of protecting the product. So it's also in the sense of the GMP requirements good that you have a flow starting with the seed lab through the main fermentation into the purification and then into the filling room, and there's no crossovers where the product is moving back into a processing room. It's just one direction of the flow, and then finally into the packaging level. The isolator, as mentioned, is closed and has a level A inside it. And that's okay because it's an isolator, and therefore the grade C in the production room is enough to provide a level A inside the isolator. When you move the vials out of the isolator, you can also treat them again with vaporized hydrogen peroxide because there is a certain risk that there is drops of liquid on the outside of the vials which could contain virus. And for biosafety reasons, if you then decontaminate them also on the outside, that makes it safer then to handle in the packaging process, and also, of course, when you send it to the hospitals, so that the people who touch the vials do not get product on their fingers. The liquid biowaste is sent down to the ground level to the kill tanks, where they're heated to, for instance, 130 degrees for half an hour, just to kill all the biological activity, and then it can be released to a wastewater treatment that is related to biosafety. Also the pressure cascades here, which means that you will always have a directed flow of air from inside inside the production rooms towards the waste corridor, so that there's no in the waste corridor, and especially when you deal with when you load the autoclave for decontamination, there will be much more open processing of liquids that are contaminated with virus, and therefore we have this pressure cascade and also this material airlocks here. So the clean team would put the used single-use equipment into the airlock, then close the door, and then the dirty team would receive it from the other side. After a few minutes, they can open this door again and then take out the used single-use equipment and bring them into the autoclave. So that's also biosafety aspects. And by the way, also all the other airlocks here, they also contribute to both biosafety and GMP because they have been designed as sinks. So the airlocks, for instance, when you enter the buffer preparation room, you will have a lower pressure in the airlock than you do in the corridor, but also in the production room. And that means that there shouldn't be any particles that are flowing from the corridor into the production room, but also the other way around should also not happen. So you protect the product by having a flow from the inside of the production room towards the outside, but you protect the operators by, the other way around. You protect the operators by having a directed flow into the airlock, and you protect the product by not getting contaminated air into the production room. So for instance, you might have zero pressure in the airlock, and then maybe you have plus 15 pascals in the corridor and plus 30 pascals in the production room, and that will give you this directed flow that you have both considered the biosafety aspects as well as the GMP aspects with the design of airlocks as sinks. In other cases, maybe for production of antibodies where the product isn't dangerous and it's not biosafety level 2, it's okay to have a pressure cascade from the inside towards the outside. So that's only protecting the product, but not necessarily the operators. But in the biosafety level 2 facility with the production of virus, you use this sink concept. Yeah, the inactivation autoclaves you mentioned, the return corridor, this is all related to biosafety, and then producing the plasmids and the infection solutions also needs to be done according to the GMP standards. I think the regulations aren't 100% clear in that respect, but I think most companies agree that, especially the production of plasmid, where you actually have the genetic sequence that defines your product, needs to be produced according to the GMP guidelines, because it's really essential for how your product is going to work later. And therefore, you have to consider the full GMP guidelines also in the production of the plasmid, even if it's just strictly a starting material of your in your process. And this is then done. Therefore, it has to be inside a production facility according to those standards as well. I think a final comment on the design of this facility is that it is a so-called outside-in concept, which means that material is moving from the outside, from this corridor on the outside, towards the inside of the facility, and then they end up sort of in the used material end up sort of in the waste corridor here. You can design facilities also inside out. In that case, you would have the supply corridor more on the inside, and you would have the return corridor, the dirty corridor, on the outside. And several facilities are designed like that. It's the same way of thinking, just the other way around. Then you have the flow from inside towards the outside. And then there are a few other concepts to do that as well. You can do everything in parallel and have sort of a supply corridor on one side, and then on the other side the return corridor. Or you can do a so-called once through, where where you just process you have the processing step from one side of the building to the other, with internal corridors that transports the product and the waste as well. So there are some different principles of designing it, but I think the most common ones maybe is the inside-out design and outside-in design. That is also a very efficient way to do it, and you limit the need for corridors because every square meter here costs money, so you would like to have as few square meters used for corridors as possible. Moving on to the cost estimates for this, and here is the capital expenditure, where we have the cost structure on the left-hand side. The building is expensive on itself. The equipment, the process equipment is expensive. And then you have your total construction costs. The professional services, this is all the engineering and the qualification works that can also be quite extensive. And then contingency and escalation. So contingency is for all the unexpected costs that you might have during project execution. They can be quite significant. If you have problems with your vendors, for instance, that they cannot deliver on time, or in the worst case, they might go bankrupt during a project, for instance, and you end up needing to look for other solutions that would probably cost you more money. Escalation is just to take into account that there's a general increase in prices. So when you plan the facility a few years earlier, the prices for everything that you have to buy during the project has increased, and so there's a 4% escalation calculated here and a 17.5% contingency also included in the estimate. That gives you this number here, 102 million of total installed cost. And then if you need to reduce those costs, and you can consider to remove certain functions of this facility. So if you take out the filling and you make it just a drug substance facility, you can save 8 million. And that is not really a big saving, and you then would lose the option of having filling in the facility, would have to do that somewhere else. You can also take out the preparation of the plasmid solution or the preparation of the infectious solution, the virus, and then you would save 6 or 5 millions on doing that. So these are the options that you have for reducing the cost of the facility, but the full scope will be then the 102 million US dollars. The calculation here was done based on benchmarks from Denmark, where there's also a biologics hub, but it has been calculated for one of the major production sites in the USA, where there's also very high costs. So there's had to be a location factor that had been to calculate the cost in the USA based on the cost in Denmark. It's a general more expensive to build buildings there. And that also goes for the cost of personnel that we will look at now. In total, there is also other factors here. Automation has been considered, but it hasn't been designed as a completely automated facility, rather with a passive SCADA that collects the data. So the cost for automation hasn't been very high in this estimate. Looking at the operational expenditures, so the production cost itself, the cost structure is the raw materials, the employee cost, wages and other costs. We have other expenses, which is roughly estimated as 30% of the staffing costs. We have maintenance costs, and then we have the depreciation of the plant, which is done with 25 years on the building part and 8 years on the processing part. This is the standard in the USA. I think in Germany it's 10 years for process equipment and 30 years for buildings. So you have to adjust it a little bit depending on which country you're building in. Or a simple way of doing it is just to take everything and assume 20 year of depreciation time. So that's how the cost structure is, and we see that 77% of it is actually the raw materials, while, for instance, the employee cost is only 7% here of the total cost. And again here, the cost of Danish employees was the basis for this calculation, and then it was added 65% to take into the account of the production cost at the site in the USA. Yeah, so that gives us an overview of the cost of production, and here finally the timeline of building this has been then estimated that you can do a 6-month basic design and then a 24-month construction phase. Before that, of course, you have to do the conceptual design. So the conceptual design is really where you design essentially what we have been looking at in this lecture. You can make the basic layouts and make the basic assumptions about the facility in your conceptual design study. Then comes the basics design, where you go into more details, and after that the detailed design, where you really design everything in most detail. For instance, you would make a 3D model of your piping, which can be very helpful, and to have a smooth execution of the project, it's very helpful to have a 3D model of the piping. Procurement and long lead items, it's here assumed that we don't need more than 12 months to order the long lead items. Filling machines often take much longer than that. It can be 18 months or maybe 24 months to order a filling machine. But here, since we're using the isolator and standardized equipment, it's assumed that this can be delivered in 12 months. And also all the other things is more or less off-the-shelf equipment, the bioreactors, the chromatography skids. These are very standardized equipment that should have a shorter delivery time than 12 months. So, but you need to start early ordering this equipment because the delivery time is so long, and it can be the rate limiting step in your product execution if you don't start early enough with the long lead items. Construction, commissioning, qualification here on this slide means operational qualification. Before that, you do installation qualification and design qualification, but really the operational qualification is here calculated with 6 months, and then we have the handover to the operator, which will be the pharmaceutical facility that is using this. And what is not considered in this timeline is the process validation, which comes after operational qualification, and it's carried out by the pharmaceutical company itself. So with those assumptions, we have a very short building time, and it's ambitious to say 30 months from start of basic design to end of operational qualification, but it should be possible. It depends on a number of factors, also any kind of problems or how well you can organize the execution of the project. It's interesting though for the company that invests that they commit at the beginning, they just commit to making conceptual design study, and that only costs a few €100,000. So that's the only risk that you do at the beginning when you do a conceptual design study. But later, when you have spent more money, you increase the risk of losing money if you want to terminate the project. And especially when you have started building it and you have started ordered long lead items, that's when you have really committed to spending those $102 million. So looking at this timeline here, there is certain decision points where the decisions becomes increasingly important then as you progress through the project. So that was an overview of how to design production plants, and we covered a lot of material. We were not able to go into details about everything, but I hope that we at least got an overview on how this can be done. And also remembering that every facility is unique. If this had been a cell therapy facility, it would have been designed completely differently. But at least we saw one example of a production facility for AAV vectors, and I hope that we were able to shed some light of some of the most basic principles behind designing facilities. So thank you for the attention.
So welcome to this part of the course where we talk about the good manufacturing practice, or GMP. Where does GMP apply to? Within the entire drug development program, GMP starts basically with the product and the process development, using QBD principles in the development to set the right control strategy and also the final manufacturing instruction and quality assurance measures. Why do we do GMP? Why it is so important that we have GMP? And the reason is that we are still having a lot of issues with recalls from the market where the GMP rules have been violated or not purely controlled on the critical attributes, so that we have contamination or sterility issues, storage and packaging issues and others. So that the problem of quality is still one of the biggest concern and also a reason for drug shortage. And if we can look into more details in this study, we see that there are a number of issues that come from a GMP or non-GMP compliant manufacturing, or issues that could have been prevented with a very strong and strict GMP compliance. So now, what is GMP, or what is a good manufacturing practice? Good manufacturing practice describe a minimum of standards that medicine manufacturers should meet during their production processes, or the overall production process from supply chain to the distribution. The European Medicines Agency is coordinating inspections to verify the compliance with the standards in Europe, with the GMP standards. And GMP combines a comprehensive set of potential quality-related elements and practices to assure that the medicines are of consistent high quality, that they are appropriate for their intended use, and that they meet the requirements of the marketing authorization or the clinical trial authorization. Now, there are a number of regulations that describe this GMP rules, and the basic rules are summarized in the EudraLex Volume 4. Here we find the basic requirements for medicinal products, and what you see here is that they are divided in different chapters. In total, we have 9 chapters, which are the pharmaceutical quality management system. It's about the personnel, the staff. It's about the premises and the equipment. It's about the documentation of what we produce and what we have to produce. It's about the production itself, how we monitor it, how we evaluate it, that it is reproducible. It's about quality control. It's about outsourced activities when we use a third party who's doing part of our manufacturing. And it's about the complaints and product recalls, because every product recall or complaint might draw us to a quality issue that we could resolve by a better control or GMP compliance. And then we do also self-inspections to verify if all the stuff, if our own manufacturing is still compliant to the GMP rules. But there are more requirements that we'll find here, for example, for the active substance used for or as starting materials, because the material which is going into a process is also part of the entire quality management system and the quality itself. So we have also here further rules and guidance on how we assure that the material is well suited. We have also a big part which deals with documents that need to be provided. For example, a site master file. A site master file provides you with the entire organizational structure, the rooms, with the entire layout. And before you get audited, you normally have to provide a site master file with all the details on your manufacturing site. But we have also the quality management system that are overlying or overarching the typical GMP manufacturing. And there are the guidances, which are the ICH guidances Q9 and Q10, that apply on a global base to all manufacturing units for pharmaceuticals. And there are also further guidelines that also talk about the health-based exposure levels, because we are also responsible for our personnel, that they are exposed to the product we manufacture, and we need to assure that our personnel is safe. And there are a number of other guidelines which apply or might not apply to a specific product, where we always look into how do we assure the right quality of the product at the time of manufacturing or development state. But we have also very specific guidelines, and as we are talking here about the cell and gene therapeutics, there is a specific guideline I would like to mention, that the good manufacturing practice is specific to advanced therapeutic medicinal products, which are more specific to this type of product manufacturing and additional GMP requirements that capture this product. Now, if we talk, or if we put this into an image, as I said, that GMP is starting already in the product development state using this quality by design guidelines. So first we need to develop a process that is reliable, that we understand that the quality and the variables are understood, and that we can monitor the process in the right way. And that is covered by the ICH Q8 R2 guideline, which talks about the product manufacturing for the intended use. We have then the Q10, which is talking about the manufacturing until the distribution. And overall, we have this quality management or risk mitigation risk management systems that assures that from the beginning to the end of the product, we have a GMP compliant manufacturing, and we control it to the extent that it is necessary to guarantee the safety of the patient as well as the quality of the product for its intended use. And what I have here on the right side, that the good distribution practice, that follows basically the GMP rules, because even the shipment of the product to the site of use is under a certain guidance, and that's the good distribution practice. Now, let's take a more depth look about how GMP evolves and what is specifically meant by it. So, manufacturing of medicinal products for human or veterinary use require GMP license. So there's no difference between human or veterinary products in terms of quality and how they should be manufactured. You need to have a GMP license, and a GMP license is product specific. You will not receive a GMP license as a general license to manufacture every product you would like at a manufacturing site. It's only for a specific product you get the license to manufacture it, according to the GMP rules and all the documentation and work you have done on this specific product. A GMP license is provided based on an intensive GMP audit by competent authorities, and normally these are the local regional authorities. In Germany, it's the Bezirksregierung der Länder, who visit the site and provide this audit, the GMP audit. And once you get a GMP license from this local authority, it's also accepted on a more or less global base. And the authorities might require the local authorities to do certain GMP audits upon request. Now, in case we do not follow this good manufacturing practice, we talk about GMP violations. And if this occurs, first you get a warning letter where they clearly describe what has to change or what they would like to see to be improved during the operation. And if we do not follow this, and if we are not replying in accordance to the expectation of the authorities, then there might be huge financial penalties and also recalls from the market of the product when it is not produced in conformance of the GMP rules. And finally, it remains the responsibility of the manufacturer, and not only the manufacturer, but GMP is a top-down approach, which means that you need to have a very strong management endorsement and support to the team to operate according to GMP. So now let's take a look into the different parts which we saw in the first slides on the EudraLex Volume 4, General Rules on GMP. There are 9 parts which I would like to show you a little bit in detail right now. So first of all, it's about the pharmaceutical quality system. A pharmaceutical quality system is a management system that covers all matters which individually or collectively influence the quality of the product. So it's everything which might happen to the product, to the staff, the environment during the manufacturing. It is the sum of the total arrangement, organized arrangement made with the objective to ensure that the medicinal product is of the required quality for the intended use. That's the sole objective of the quality management system, that we control it or monitor it throughout the process. It's about the size and the complexity of a company's activities that should be taken into consideration when developing new pharmaceutical quality management systems or modifying existing one. As I said, the GMP is given for a product-specific or is a product-specific license we get. And if we develop additional products, different products, they might be interference, so we need to adapt or modify the quality management system. And that's why we also need to really take into account the complexity of a GMP manufacturing site. And there's no standard quality management system that you can put over every manufacturing site. And then we have the pharmaceutical quality management system, which is incorporated in the good manufacturing practice, and the quality risk management. The next one is the personnel. What do we mean here when we talk about GMP and personnel? First of all, it is a senior management is ultimately the responsible one and has to ensure continuously that the suitability and the effectiveness of the quality management systems and the GMP compliance through participation in management reviews. This is, I think, a very important message, that the management cannot say, oh, I didn't know what happened down in my manufacturing. They are responsible and they have to provide the resources that the staff can work in compliance with good manufacturing practice. It requires at least 3 independent people. It's the head of the production, it's the head of the quality control, and finally there need to be an adequate number of what we call qualified persons. These are experienced people on a certain product who are able to release the product according to the specification and the intended use, and also and also support that the manufacturing was done under GMP and that the quality of this specific batch of the product is according to all the required specification. And key posts should be occupied by full-time people, full-time employees. And we always need to educate these people. They need to be present and survey the facility all over the time when it is operating. And the heads of the production and the quality control must be independent from each other. Otherwise, there might be a bias in interpretation of results or interpretation of manufacturing compliance. And the people in responsible positions should have a specific duty recorded in a written job description and an adequate authority to carry out the responsibility. So the idea here is that everybody understands the responsibility they have and that there is no possible bias that they said it was not me who was responsible, I thought that my colleague. Everything is written down, and they have the authority to stop a manufacturing or to not release a product without being punished by anybody. So that is a very important role of the personnel, that we know who is doing what and what they are responsible for and what is their duty. Besides this, we have the premises and the equipment. So the premises and the equipment must be located, designed and constructed, adapted and maintained to suit the operations they should carry out. Which means whatever we bring into a manufacturing process in the premises as an equipment need to be validated, qualified for the purpose it is used, and need to be installed in the GMP premises. The layout must permit that we have an effective cleaning and maintenance in order to avoid any adverse effects or quality issues of the problem. That, for example, accounts for if you have microbiology issues and if you have not surfaces which you can clean completely without having some pockets where microbiology could grow, then the layout is not appropriate. And you should not use any material which you cannot sterilize, for example. So this is a very important part of designing a GMP premise and equipment installation. Then about storage areas, there should be sufficient space starting from packaging materials, intermediates, finished products, products in quarantine, released products, rejected, returned products or recalls. Because we need to make sure that we can trace it back and that we have also the qualified material for manufacturing included in the manufacturing sites early on. Then control laboratories should be designed to suit the operations to be carried out in them. There should be adequate storage space for samples or any records we do on the samples. So the manufacturing itself need to be supported by a good quality assurance laboratory space very much aside. And then we talk about equipment that should be designed, located and maintained to suit the intended purpose. And this also includes that we do repair, maintenance, cleaning of the equipment close by and continuously. So the fourth aspect of the GMP is the documentation. The documentation has to establish control, monitor and records of all the activities which directly or indirectly have an impact on all aspects on quality of the medicinal product. So the important aspect here is that we are able to trace back where materials are coming from, what happened to the materials, and what could happen unexpectedly to the product. It's also that we have documentation that may exist either in paper or electronic or photographic media. Everything is possible, but the documentation need to be solid. You should not be able to modify retrospectively any of the documentation to change some data or some information that you gathered during the manufacturing. And then there's a site master file. This is a document that describes the GMP related activities of this manufacturing site here. And then we have the record and the report type. We have a generation of the control documentation, good documentation practice, retention of documentation, specifications of the starting materials. We have batch processing, packaging records, material receipts, and samples and testing. And everything is laid out in this master file, how we deal with it, what we have and what we do. Finally, it comes then to production. What do we do in production? And the production must follow a clearly defined procedure complying with the principle of GMP to obtain the product of quality in accordance with the relevant manufacturing license. Which basically means that within any manufacturing campaign, I have a clear procedure which I have to follow, including all the controls or monitoring exercises I do, the sampling exercise that need to be assured during the manufacturing. Then the production should be performed and supervised by competent people, which means that I do not leave people alone just, doing it unless I have some kind of surveillance that they work according to the GMP and the manufacturing instructions. Quality risk management processes is the basis to determine the extent of technical and organizational measures required to control the risk. This goes back to the fact that in a manufacturing, we identify early on in our development that there are certain aspects, either material specification or process parameters, which bear a risk of failure in the final quality, which means that we need to have a clear risk management system, how we would deal with certain risks and how we avoid that these risks get through. Then we have a validation studies should be reinforced GMP and conducted in accordance with the defined procedures, and the results and conclusions should be recorded. So we always, do some validation studies if the manufacturing is still within the quality range and the GMP rules. And any deviation or unexpected observation has to be recorded, investigated, and possible samples to be taken. Which means that after the manufacturing campaign, I might have some observations. To judge the observations on the impact of the quality, I need to document them. I need to record, investigate what happened, why it happened, and also having some samples that I could analyze the intermediate and see if I have to take an action or if we can still release a batch. Quality control is an independent quality control from production is considered fundamental to have a satisfactory operation of the quality control. So as I said, manufacturing without quality control will never happen. So there is a specific emphasis on the quality control. And the quality control is concerned with sampling, with specification, with testing, with organization, documentation, and release procedures which ensure that the product is released until the quality has been judged satisfactorily. And the quality follows the principle of good quality laboratory practice, which is another set of guidance or rules that the quality control itself is valid and correct for the purpose I use the quality control. And then quality controls also include ongoing stability to monitor the product over the shelf life and to determine that the product remains within this expected quality range and the specification we have set. So we are not using quality testing or quality controls to test quality into the product. It's the other way around. We use quality control just to design quality into the product. So that means we have a clear understanding where we have some quality issues or might have quality issues, where are the risks, and we design the process from the start by the Q8 guideline on quality by design to avoid that we are just doing some tests to confirm quality, but we know that the quality is achieved by the process and we just monitor it. Outsourcing activities are something which is often being used in manufacturing of pharmaceutical products, as we might not have everything in-house, so we use external partners. But external partners are part of this entire GMP as we ourselves in our facility are. So any activity that is outsourced should be appropriately defined, agreed and controlled to avoid any misunderstanding, what responsibility or what specific role an outsourcing partner plays within this GMP. There must be a written contract between the two parties that clearly establish the duties of every party. And the quality management system is conducted, is a contract giver, must clearly state the way that the quality person certifies each batch and product for a release exercise and this full responsibility for it. So also the way that they deal with the quality and the quality management system of the outsourcing partner must be compliant with GMP, and we need to be able to trace this GMP compliance of the outsourcing partner back. The contract should describe clearly who undertakes each step and of the outsource activity. It's again about the responsibility, very clearly defined who is doing what and who is responsible for it. And any contract acceptor should be considered and must be audited. So do not rely on somebody who is telling you, I sent you over the document, we did it in accordance with GMP. At a certain moment you have to audit the supplier to assure that their procedures and their manufacturing is also in accordance with the standards of GMP rules. Then we have also compliant quality defects or product recalls. Whenever we are in a manufacturing environment, it might happen that something in the product or in the manufacturing occurs that we do not really like. So what we have to do is all the concerned competent authorities should be informed in a timely manner in case of a quality defect, just for information first. Then we take the complaint, we document it, and we assess it to establish if there represents a certain quality defect or any other issue that requires an action from us to be taken. Then we appropriately train any experienced personnel to be responsible for managing the complaint and the quality defects is able to decide on what measures need to be taken to manage potential risk or any of these issues that might go up to a recall of the product to protect the patient. And an appropriate level of root cause analysis work need to be done. And we need to investigate why this quality defect occurs, because this gives us also the possibility to modify our quality system or our manufacturing process to avoid that this defect, quality defect, occurs another time. So it's a lot of paperwork, and each of these complaints need to be treated in the same way. And this is a logic sequence. We take it, we document it, we analyze it, and then we take the measures either to the batch manufactured itself or our manufacturing processes and controls. And the last thing is the self-inspection. Self-inspection means that we do internal audits that should be performed on interval base, either by ourselves or we use an external consultant to do it, to reconfirm that our personnel works according to GMP, and also helps us to identify if additional training or anything is required. We have to record these trainings. There need to be a report that contains also all observations being made during this inspection, including that the proposals for corrective measures. Because if you work routinely in a manufacturing, you might get habits, take shortcuts which are not in accordance to the manufacturing instructions or the GMP rules. And this can then be corrected by training or by re-emphasizing the correct measurement or the correct handling of the product. So that are really all the aspects. So in conclusion, what GMP is. GMP is really a science of manufacturing consistent quality. Materials and process science is included, and it covers the entire manufacturing cycle, from the raw materials, from the supply chain up to the distribution into the sites of use. GMP is a global standard for medicinal products for human use and veterinary use. So it's not something which is just regional, that we are very strict and others are not. No, this is a global standard of manufacturing of medicinal products. It is directly linked to the quality, and the quality is linked to patient safety. So it's all about being able to reproduce the same quality and the quality that we have intended, that we have specified for the intended use in our approval, drug product approval. And it's honestly a mindset that becomes a routine once you start to practice it, because it helps us to really also understand how we operate and our influence, and the entire manufacturing can be guaranteed, or the quality of the manufacturing can be guaranteed by this guidance and this behavior that you get learned once you manufacture or once you are in a GMP environment. And it is an obligation and the responsibility of the manufacturer and the management of this manufacturer. So that is important to say that if you are working in a GMP and you see that you have some issues, then you need, or the management has to provide you with what is necessary to be compliant and manufacture the same quality over and over again. And this were my last words here, and this was a short introduction in GMP, and I thank you for listening.